So, hello everyone. Welcome once again to the What We Are Reading YouTube channel of the South American Mycorrhizal Research Network. I am César Marín, and today we have the pleasure of having with us Olga Lastovetsky, if I am saying your name correctly. She has recently been appointed as an assistant professor in the University College of Dublin in Ireland, and she will be talking with us about her recent study in new Phytologist, just recently published, regarding the high diversity of endobacterial um, taxa in arbuscular mycorrhizal fungal spores, a very interesting topic. And I want to start by giving the definition that is in the paper of endobacteria. Endobacteria in this context are defined as those bacteria that exist inside the arbuscular mycorrhizal fungal spores, either directly in the cytosol bound by host compartments or embed in the spore wall. So my first question to you is how this study came about? Is this part of maybe your postdoc or is based on previous research? To be honest, the only paper that I remember about this is by Paola Bonfante from 2004, like showing the vertical transmission of this bacteria. So how this study came about?
Yeah, so I actually became interested in bacterial fungal interactions during my PhD work. I worked specifically on fungal endobacteria. And at the time, fungal endobacteria were not really well known. They weren't really well characterized. They're sort of thought to be these biological oddities. But since the early 2000s, when really the more seminal work on fungal endobacteria came out, we're sort of now realizing that a lot of fungi that we previously thought were sterile actually harbor endobacteria. And so one of the projects that I worked on was on the endobacteria of arbuscular mycorrhizal fungi. So arbuscular mycorrhizal fungi, or AMF, they have actually been known to harbor endobacteria since the 70s, when work by Barbara Mossy discovered these, what they called bacteria-like organelles inside AMF spores and hyphae using electron microscopy. And then, like you were saying, with Paola Bonfante's work, she began to characterize these bacteria. She was the first one who actually proved that these were bacteria. And she described two different endobacteria that can exist and can coexist in arbuscular mycorrhizal fungi. So they are called, one of them is called Candidatus. Actually, maybe I'll share my screen and I'll show the pictures at this point.
Yes. Yes.
Let me do that. It's helpful to have pictures when you talk about
Excellent.
complicated systems. Okay, so the two known endobacteria that have been described in AMF are called, so here's one, Candidatus glomarbacter gygospirarum, because it's a bit of a mouthful. I kind of call it glomarbacter when I give a presentation, or in the paper, they're referred to as CAGG. So CAGG or glomarbacter, they are a little bit better studied. So Paolo, mainly work from Paolo Bonfante's lab, they've shown that they actually are mutualists, or they have characteristics of mutualists and of being mutualists to AMF. So they appear to help prime the energy metabolism of AMF. The isolates that have glomarbacter appear to have increased hyphal proliferation during spore germination versus isolates that don't have glomarbacter. And also this glomarbacter appears to increase the sporulation success of AMF. The other known endobacteria of AMF are called Candidatus moeniplasma glomeromycotorum, also a bit of a mouthful. I'll just call it moeniplasma. And in the paper, they're referred to as CAMG for short. So moeniplasma, the role of moeniplasma in AMF biology remains experimentally uncharacterized, which essentially means we have literally no idea what they do in AMF or in the interaction between AMF and the plant. But there's a good, so we know that the genome sequence of moeniplasma, and there's been some molecular evolutionary analysis done on these endosymbionts, which indicate that they have potentially characteristics of parasites. But essentially, we don't know what they do. And so when I began working on this AMF endobacteria relationship, everything we knew about AMF endobacteria came from studies of AMF derived from culture collection isolates. And nothing was really known about this relationship in nature. So during my PhD, I looked at the distribution of these endobacteria in natural AMF populations. And we did this study by isolating AMF spores directly from the field. In this case, this was a natural dune ecosystem located on the east coast of the States at Cape Cod in Massachusetts. And from this sampling site, we extracted AMF spores and then we surface sterilized them. So we surface sterilized and made sure that we're really looking at everything that's on the inside. And then using molecular techniques, PCR with specific primers to known endobacteria, we discovered that the Glomarbacter endobacterium, the one that we know what it does, it's a mutualist, was actually very rare. Whereas mycoplasma, the one that we don't know what it does, was actually very, very common. So the Glomarbacter was found in only about 2% of the spores, whereas mycoplasma was found in almost all of the spores.
So, can
Yeah,
I interrupt for a second? This makes me wonder, how do you experimentally prove their role of, for example, mycoplasma?
exactly. Yeah.
What kind of experiment would be necessary to know what they do inside the AMF?
Yes, a very good question. So really, and how this is done with other fungal endobacterial systems is you really need to get isogenic or genetically identical isolates, in this case, say, of AMF, that differ in the presence and absence of, in this case, mycoplasma. And then you can start comparing their, how well they germinate, how well do they grow, how well do they associate with the plant host, what are the differentially expressed genes in the AMF versus in the plant host. So all of these things have not been done yet because nobody has been able to cure the AMF, quote unquote, from their mycoplasma endosymbionts. But once this is actually something we're trying to do in my lab at the moment, we're trying to obtain these isogenic mycoplasma plus, mycoplasma minus isolates and try to understand their function in the AMF and in the plant AMF interaction.
Okay, thanks.
So the reason I'm telling you this now, the other thing that we found in this study, this was published back in 2018, is that we sort of detected a potentially third different kind of endosymbiont in AMF. But this was very almost like a side story. And this came about because our Glomarbacter specific primers weren't very specific, and they ended up amplifying other sequences that were more closely related to, the relatives of Glomarbacter, which are Burkholderia. And so this can be seen from this tree in that, so in red and in blue are sequences we amplified from the spores from the field. And you can see some of them group with Glomarbacter, so with the known endosymbionts, but others group with these free-living Burkholderia. And so we kind of concluded, oh, it is possible that maybe there's this unknown Burkholderia-related endobacteria that can be found in AMF. So this was sort of setting the stage for the current study that we're discussing, where I really wanted to look more closely at first of all, the distribution of AMF of endobacteria in natural AMF populations, other than just one site, but also try to characterize all of the endobacteria that could be associated with AMF spores. Mm. And so this work that we're discussing in the new Phytologist paper, this was done when I was a postdoc. And so this was kind of after I did my PhD in the States. I moved to Ireland and I wanted to continue working on the system. So again, we did some field sampling at 2 nearby sites on the east coast of Ireland now. We looked again at a natural dune site and then a nearby agricultural grassland site. So from these sites, we again extracted AM fungal spores, we surface sterilized them, and we first of all checked for the presence of the 2 known endobacteria. And what we found was, I think, was quite remarkable, is that just like at the Cape Cod dunes, thousands of miles away, the Glomarbacter endobacterium, the one that's a mutualist, was actually very rare, whereas the mycoplasma endobacteria, the one that we don't know what it does, but maybe it's parasitic, was again extremely common. And the kind of rate of distribution of these 2 endobacteria was almost identical to what we found at Cape Cod in the States. So one of your kind of what we're thinking about discussing is what does this pattern mean?
Yes.
Because it appears to be similar in all of these ecosystems, which differ in soil type and dominant plant community type. Some of them are thousands of miles apart. And the short answer is that we don't actually know.
This is so
But
interesting. Yeah, sorry, go ahead.
no, but I guess I guess in order to begin to know, we, like you were saying, we need to understand what the role of these endobacteria, particularly the mycoplasma endobacteria, is in AMF, to
Mm.
try to start understanding why are they so prevalent.
This reminds me of a study by Gu Feng in China about the ionosphere. Uh,
Hmm.
we made an interview before to him, but they also found the same bacterial gills in the ionosphere in three different, very different ecosystems across China. So it's always remarkable with these bacterial and fungal systems, this kind of, yeah, the stability, it seems like the proportion of these of both taxa
Hmm.
is stable, and also there is a 19%, I guess, of other totally different unexpected bacterial taxa. Maybe can you talk about a little bit
Yeah.
about this?
Yeah, absolutely. So that's a nice segue to what we did next. So next, I wanted to characterize all the endobacteria associated with these spores. So we had these 84 AMF spores, which tested positive in a PCR-based screen for the presence of any kind of bacteria. And so we then subjected them to bacterial community profiling using 16S amplicon sequencing on the Illumina MySeq platform. And what I found was very unexpected. I found that the communities associated with these spores are very bacterial communities associated where living inside these spores are very diverse. So what you're looking at here are plots showing the relative abundance of bacterial phyla in the AMF spores. So this graph in the middle is just splitting it by two different sampling sites. And the graph that's on my left is showing each of the kind of, on the y-axis, you're seeing individual AMF spores, so all of the 84 spores. So you can see, first of all, that first of all, you can see that they're almost all of them are red. And in red are the bacteria from the Tenerocutis phylum, and these are the mycoplasma endobacteria, the CAMG endobacteria. So you can see that they're the most abundant inside, they have the highest relative abundance inside spores, but they also seem to be the most prevalent. So almost all of the spores have the mycoplasma, which is what we talked about in the previous slide. The other endobacteria, known endobacteria, Glomeribacter, CAGG, they belong to the Proteobacteria phylum. However, the Proteobacteria sequences that you can see shown on these plots are not even Glomeribacter. Glomeribacter was so rare, I had to really dig to find them bioinformatically. These are other Proteobacteria, mostly gamma Proteobacteria. So things like, Escherichia coli, they're gamma Proteobacteria. But again, another thing that you can see from this plots is that, there are bacteria belonging to 10 other phyla that are not, Mycoplasma glomeribacter that can be found inside these spores. And so, in fact, in an individual AMF spore, we could detect up to 277 other bacterial, OTUs, which is a proxy for bacterial species, with a mean of about 25 other bacterial OTUs, the ones that are not the known CAGG, CAMG endosymbionts, with a mean of about 25. And so it wasn't like 80% of spores had mycoplasma, 1% had glomeribacter, and the rest had these other endobacteria. These other endobacteria could be detected in spores that also had glomeribacter and also had mycoplasma. And sometimes glomeribacter, no, and actually at all times glomeribacter and mycoplasma co-occurred together in individual spores. Sometimes there were other endobacteria, sometimes there weren't. So this was, I think that was pretty cool.
This is so interesting. Yes.
So then, of course, as what are these bacteria? I'm trying to understand if there's any pattern. So first of all, I couldn't detect any particular pattern. I was thinking, oh, maybe I discovered a third different kind of endosymbiont, a fourth, a fifth, but I
Yeah.
couldn't discover any particular pattern. There wasn't like there was one other bacterial taxa that was present, in multiple spores.
Mm.
And in fact, all of these other endobacteria seemed to be related to bacteria with known free living lifestyles. So such as, bacteria that are known to be free living in the soil or free living in aquatic environments. Some of them were facultative symbionts of plants. I detected rhizobius sequences there, for example. Some of them were, opportunistic pathogens of humans or other protozoa. So there's no clear pattern. But what I kind of, what we sort of started with, what I started with the study is that, we know there's the plant, inside the plant roots, there's the AMF, and inside the AMF, there could be two bacteria, but it seems actually inside AMF, at least spores, there could be whole communities of bacteria. And we still have no idea what they do.
Wow. And you talk because you talk in the discussion about this conditional mutualism. Uh, so which of these three types of bacterial taxa that we have seen so far would be the responsible of this conditional mutualism, and also under which conditions would be the, because we know that for AMF and plant systems, for example,
Mhm,
like limited resources, enhance mutualism, and in this
mhm.
case, which conditions would enhance the mutualism between bacteria and AMF?
Yeah, that's a very good question. The short answer is that we don't know, but I can just tell you what it is that I think. So first of all, I started thinking about what are all these new, whole communities of endobacteria, could they be doing in AMF spores? And at the time that I was analyzing this data, there's this really cool paper that came out by Venkatesh and colleagues from Nancy Keller's lab, where they showed that in other fungi, they were working with Aspergillus.
Mhm.
The chlamydospores, or these like really thick-walled resistant structures of these fungi, they could be invaded by free-living bacteria, and that being inside these fungal chlamydospores appears to protect these bacteria from abiotic stresses such as low nutrient stress or low temperature. So the ones that were inside the spores, the bacteria that are inside the spores in this study were able to survive when exposed to cold and nutrient stress, whereas the bacteria that were outside were not able to survive. So in this study, they called them bacterial hitchhikers. And they hypothesized that being able to invade these fungal spores could explain the persistence of certain bacterial taxa under harsh environmental conditions. And really, this is quite phenomenal if you really think about, how do bacteria survive under, really, really cold winter conditions in the northeast of America, for example. I guess this is one of the potential explanations for at least some of these taxa. They can overwinter in these nutrient-rich resistance fungal spores. So I think when I was reading the study, I was like, maybe this is what is happening in my with my AMF spores. So we kind of based on this, we've developed this hypothesis that maybe these diverse bacterial communities inside AMF, they are also composed analogously of these quote unquote hitchhikers.
Yeah.
And this is not so hard to believe. I mean, AMF spores are packed full of delicious lipids and glycogen that could serve as nutrients for these bacteria, and the spores that theoretically could also protect them from these harsh conditions. So that's one of my hypotheses. And um, then you wanted me to talk about the other, the conditional mutualism.
Yeah.
So to talk about the conditional mutualism, we have to now kind of switch gears and talk about the CAMG, or the Winnie plasma endobacteria, the one that's a bit less studied, the one that we actually have no idea what they also still do in AMF. Now, they are a little weird in that their populations, so the Winnie plasma populations, can be highly diverse, even within individual AMF spores, with at least 3 different bacteria and, Moenoplasma quote unquote species or OTUs have been known to coexist inside even an individual AMF spore. And incidentally, Glomeribacter populations are always homogeneous or uniform inside individual AMF spores. So in this study that we're talking about, using deep sequencing, we discovered that an individual AMF spore can be home to as many as 50 Moenoplasma OTUs, or quote unquote species, with an average of 22 OTUs per spore. Now, this really high genetic diversity of endosymbionts is usually taken to be a characteristic of parasites because the diverse endosymbionts are expected to have higher competition dynamics within an individual host, and therefore could be more detrimental to the host. And actually, being highly diverse inside individual AMF spores is one of the reasons why Moenoplasma were hypothesized initially to be parasites. But like we've been talking about, Moenoplasma or CAMG, they're extremely common in AMF isolates, with about 80% of all of the spores that we've surveyed so far from the field harboring them. So I think that it's very unlikely that Moenoplasma will be maintained to such high levels in an AMF if they don't provide any benefit to their host. So based on this, we are hypothesizing that maybe they're conditional mutualists. And in a conditional mutualism, endosymbionts provide a benefit to their host under specific conditions. And so in the realm of bacterial fungal symbioses, endobacteria are known to provide a conditional benefit to their fungal host in the presence of predators. So these are 2 really prominent examples. So there's a symbiotic system. There's a fungus called Rhizopus microspores, and they harbor bacteria called Myceta habitans rhizoxynica. Myceta habitans rhizoxynica, they produce a toxin that protects the Rhizopus host from soil micropredators such as amoebae and nematodes. Analogously, in the symbiosis between Mortierella fungi and their Mycoavidus endobacterial symbionts, the Mycoavidus endobacterial symbionts also produce a toxin that protects its Mortierella fungal host from, again, predation by nematodes. So in this case, these bacterial endosymbionts provide a benefit to their host in the presence of predators, and they would be called defensive mutualists. So I'm hypothesizing that Moenoplasma analogously could be viewed as defensive mutualists of AMF. And this hypothesis came about because of this observation that I made when I was looking at the population structure of Moenoplasma versus the community structure of all of the bacteria. So what I found was that inside individual AMF spores, as the Moenoplasma diversity, or as the diversity of Moenoplasma populations increased, the diversity of all of these other non-Moenoplasma endobacteria decreased. So in this plot, every dot represents the diversity of the community inside AMF, an individual AMF spore.
Yeah.
So
But
basically,
this
sorry.
is sorry. In the y-axis is also excluding Glomeribacter or including Glomeribacter?
That's a very good question. The y-axis, I have to think about this. I think it might be including Glomeribacter, but Glomeribacter was so rare that it is unlikely that it would do anything to influence. It was like, we can talk about Glomeribacter in a minute. It was so rare, I had to like fish for it in my bioinformatic data. Yeah, there's only 3 Glomeribacter species that were taxa that I found in all of the spores. Okay. But anyway.
Yeah, sorry.
Yeah, so I guess my point is that I think that by maintaining this high population diversity inside AMF, the Moenoplasma endosymbionts are likely more effective at outcompeting all of these other potentially harmful or these hitchhiker endobacteria. And so by kind of tolerating highly diverse populations of Moenoplasma inside itself, an AMF fungus would still just be home to one type of endosymbiont versus, whole communities of endosymbionts. Um,
So
yeah,
it's
of course. Yeah.
sorry. So it seems to me that like let me ask you
Mhm.
if the physiological or biochemical mechanisms by which these bacterial cells enter the spores, is this known how they actually enter? No?
No, it is completely unknown. So, well, no, the glomerobacter and the mycoplasma likely enter from the same way that the nuclei would populate the AMF spore. They will be carried with the, the cytoplasmic flow, the protoplasmic. They will be basically populate the spore from the hyphae.
Yeah.
Whereas how these other, quote unquote, hitchhiker endobacteria enter the spore is completely unknown. But we know that this is done in other, in these in Aspergillus in the work from Nancy Keller's lab. So it must happen. Yeah, the short answer is we really don't know, but I'd love to find out.
But it also seems that there is vertical transmission going on, right?
So vertical transmission going on of definitely the known endosymbionts. So mycoplasma and glomerobacter, they are vertically transmitted. But but we don't know whether all these other endobacteria are, do they populate the spore as it's being formed? Do they populate the spore after the spore is already formed?
Mm,
For somehow digesting the spore wall and entering through it, for example?
mm.
Or do they populate the hyphae first and then become vertically inherited in the spores? That's completely unknown.
Okay, that's so interesting. And can you just to finish talk a little bit more about the glomeribacter, why they were so not abundant, and if it's possible that their mutualism is also conditional or they are just mutualist?
Why are they so rare? I don't know.
Okay.
It could be that, well, the glomeribacter is kind of has been up until this study known to be just an endosymbiont actually of the Gigasperaceae family, of the AMF from the Gigasperaceae family.
Mhm.
Um, and it is mainly, and even in my study, it's mainly found within the spores of Scutellaspora. Although there is one or two spores of Paraglomus that also I seem to have detected sequences belonging to Glomeribacter from those spores. So it could really be the reason they're so rare is because the I guess the kind of fields or the field study sites that we've chosen that maybe had less of the community of the Gigasperaceae, although that was one of the reasons why we chose dunes, why we chose dunes in the first place during my PhD is because they're known to be Gigasperaceae are known to be in high relative abundance in the dunes. So we were kind of trying to maximize that chances of finding these endobacteria.
Mm.
Um, how this low prevalence questions whether it questions their mutualistic or their benefit to AMF, I cannot really tell you, but I cannot really speculate as to why they would be so rare, even though they're potentially they have potential benefits. Maybe they're not as helpful as mycoplasma under environmental conditions. I don't know.
Well, thanks a lot for this very interesting interview. There are several more questions that I wanted to ask, but I encourage our listeners to just go to the paper as always and check because there are many more analyses about, for example, the drivers or the factors structuring the communities of this endobacterial taxa. They also have a very nice phylogenetic tree of the AMF analyze. So please go and read the article. And I wanted to finish asking you, what are the next steps? What are you currently doing, both regarding this really interesting research area, but also professionally?
Yeah, so currently I have two PhD students who have just started in this past September. And the kind of the main push, one of the main aspects of my ongoing research is trying to understand functionally what the Plasma endobacteria are doing in AMF and how this impacts the AMF plant interaction. But in general, my research and what my research interests are fungal bacterial interactions, particularly endobacteria fungi. And endobacteria fungi appear to be more common in the early diverging fungi. So that's one of the, I'm interested in other early diverging fungi, such as, Mucormycota, Mortierella mycota, and their bacterial endosymbionts as well, and kind of functional roles in the host, molecular level interactions, evolution, ecology, all those cool things. Did I answer both your questions?
I think so, yes. And I want to finish just by saying that it seems to me like all these bacterial findings inside the spores, in the hyphae, all what they are doing in the hyphae. And I also think that for ectomycorrhiza, many people is finding a bunch of roles of bacteria in ectomycorrhizal symbiosis. So yes, just put way more attention on what bacteria are doing, and maybe this push us to maybe not redefine, but make a way more complex definition of what a mycorrhiza is, because this bacterial taxa seems so prevalent, so everywhere that, yeah, we cannot ignore them anymore.
Exactly. I very much agree, Cesar.
Okay, thank you so much, Olga. It was a very nice interview.
Thank you so much.